It's very exciting to be here. Thank you very much for the invitation. When Eleazar invited me to come to this meeting, I said, oh, I'd given a talk here about 6 months ago. I said, can I give a similar talk? He said, that'd be a great talk. I think he didn't realize I'd be giving the same talk. And so, a part of that is United's fault. My flight got in late, and so it's never a good idea to apologize at the start of the talk, but now that we've gotten that out of the way, we can just have fun, right? And so my name's Quaid Morris, and I'm at the University of Toronto, at least for the next 3 months or so. I'm about to move to Memorial Sloan Kettering Cancer Center in New York. And as you can see here, I have a lot of affiliations, and that's one of my hobbies, is picking up affiliations. At one point, I was a member of 3 different faculties at the University of Toronto, and I had students in my lab from 5 different departments, and I was trying to figure out what the 4th faculty I would get would be. It would be pharmacy or nursing, or also I had like public health in mind. But then I learned an important lesson for all the junior faculty out there is that different new departmental affiliations, they just give you further responsibilities. But if you get affiliations outside the university, that gives you money. So now I'm affiliate of the Vector Institute for Artificial Intelligence, and I'm a member of the Associate Investigator of the Ontario Institute of Cancer Research, and I would like to thank them for funding my research. And so today what I'm going to talk about is, I give a kind of broad thing, a field guide to artificial intelligence and cancer genomics. And I see myself as a machine learning researcher. We use the term artificial intelligence now when we want to get money. And that's another important thing to keep in mind. That's going to be good for about 2 more years, and then we're going to have to go back and we'll switch to another term. And so, when you are a machine learning researcher in like a biomedical field, it's very hard to find kind of the perfect problem that you want to work on. And the perfect problem is one that has sort of an interesting methodological challenge, so you can still say interesting things to your machine learning friends, but also it will have some impact on biological questions that you're interested in. And so you spend a lot of time thinking about what would be, not only like what would be an interesting question to answer, but what is a sort of a field that I can move into that's going to give you a lot of like further methodological research going forward. And so what I want to talk to you about today is some of the things that we've been thinking about and working on recently related to the analysis of somatic variants. And I think we have a slightly different take on it than the normal take of somatic variants. And I think that the way in which we're thinking about the problem and other people are thinking about the problem is a fertile ground for methodological research of the type that will actually answer interesting biological questions. Okay, so first, what is artificial intelligence? And so when you think about artificial intelligence, people, they give you a Venn diagram that contains computer science and statistics. And the question is, well, where do you live in this Venn diagram? Does anybody want to say? Well, I asked my student this question, and she said, where are we, Quaid? We're everywhere. That joke worked a lot better 6 months ago. And so what she really meant by that is like, not only do we include computer science and statistics, and if you do either of those 2 things, you can call yourself an artificial intelligence researcher right now. So you're all artificial intelligence researchers, congratulations. But also to do our type of work, you need to have some sort of domain knowledge, right? And so what do these different fields give you? Well, from statistics, you get the models. This gives you kind of a principal way of analyzing data, so you know what you're doing when you're fitting models to data. And computer science gives you the algorithms, right? If you don't have good algorithms, you can't even fit models to data. In fact, some types of models are going to find local minima, and without good algorithmic knowledge, you won't be able to identify where the global minima are, or you'll run out of time to fit your model. And the last thing you get for the domains, you get some sort of meaning, right? And so you really want to combine all these strings together when you're doing computational genomics, because that's where your science is going to be, especially if you're a dry lab researcher like we are. Okay, and so as I pointed out, I'm at the point of size in my lab where I have to hide the size of my lab, not inflate the size of my lab. So here's my lab, and we work largely in cancer genomics, though we still have a large interest in gene regulatory networks. That's where we started from, in RNA binding proteins. And in doing machine learning, people show up with interesting data sets all the time. So we've been working a little bit in electronic medical records and other types of clinical data. We've been doing a lot of work on image analysis, and we've just started to do some interesting work in metabolomics. I'm here all week, and I'm happy to talk about any of these topics with people. And so we're going to skip a few slides here, because I've got 45 minutes. OK, so let's have a question. Let's get people activated. Does anyone want to offer an answer to this question? People who haven't seen me present this slide before? Especially people who have seen me present this slide more than 10 times before. How many DNA mutations do you accumulate in your lifetime? We're talking about somatic mutations. So things that aren't in your germline, things that aren't shared by everyone in your cells. Is anyone willing to go as high as 1 billion? It's a big number. Yeah? OK. There's 2 people willing to go as high as 1 billion. Anybody willing to go as high as 100 billion? No. OK. 100 billion? All right. What about 1 trillion? That's a big number, too. Can you double count? Yeah. So that's a good question. So there's actually only 10 billion possible mutations to your genome. Right, so if you're going to go above 10 billion, you got to allow the same mutation to happen more than once, and we will allow the same mutation to happen more than once. Okay, so I think the number is a quadrillion. And why do I think that? Well, the first thing is there's 10 trillion cells in your body. And these 10 trillion cells in your body that have DNA, that aren't red blood cells. Right, and so of those 10 trillion cells, you get 1 or 2 mutations per cell division. That's under ideal conditions, right? So everyone's born with 50 new mutations. And if you do the math, that kind of works out to sort of the minimum number of cell divisions you can go to from like the germline to the embryonic stem cell. But most of the somatic cells in your body, they divide multiple times, right? So a cell divides like once per year, let's say on average. And we're looking at already 10 trillion times 100 is already at a quadrillion. That said, there's some cells in your body that divide a lot more, and certainly a lot of people do things that increase their risk of cancer, like smoke. Right? So if you put all those things together, you're looking at somewhere between 50 and 50,000 somatic mutations in a healthy cell. And so, to give you some more data, if you sequence primary tumors, samples of primary tumors about 30 to 50x, so you read the genome 30 to 50 times and then count the number of mutations you see about 5,000 detectable mutations in that sample. Okay. So what does that mean? Well, because there's only 10 billion possible mutations, every possible mutation will happen at least 10,000 times somewhere in your body. So if there's one somatic mutation that causes cancer, we're going to have somewhere between 10,000 and 100,000 different cancers going on simultaneously at the same time. So why does this not happen? And obviously the answer to that is it's not a single mutation. It's multiple mutations. People estimate the number somewhere between 5 to 10 important mutations happen to transform a normal somatic cell into a cancer cell. And so what do these mutations provide? Well, these mutations give cancers kind of a shared set of traits. And so maybe before I get into the slide, let's just step back and let's talk about what cancer really is. I think of it as an infection by like an organism that's specifically designed to target you. It's a single cell organism that just happens to be related to us. And in fact, it's kind of weird to call things cancer because what we're talking about is we're talking about a collection of single cell organisms that are distinct in every single person's body. Luckily, humans don't have transmissible cancers yet, but maybe we'll get one sometime in the future, or hopefully not. If we do get one, its name will probably be Gila. And so it's kind of amazing, actually, that we do see any shared variants at all between cancers. Because the only thing that's similar among them, except for the fact that they're colonizing our bodies, is that they have a shared ancestry, and that shared ancestor is us. Okay. So nonetheless, despite this kind of to me seems kind of a mind-blowing thing that these things are shared. They tend to acquire these common traits. And this is a very classic picture that you'll see, and this is called the hallmarks of cancer. They were originally defined in 2001, and there were later ones defined in 2011. And they're all phenotypes that successful cancers collect. And so presumably the driver mutations that are necessary to do the transformation are the ones that are providing these phenotypes. And so people have started analyzing cancer genomes as though they're fossil records. And so why is that? Oh man, it sucks to not be able to point. You guys can't see it if I do that, right? No. Yeah, there we go. Okay, I don't have the thing that, oh, there the mouse gets bigger. All right. So, because the way the cell division works, we just accumulate, our cells, our somatic cells, accumulate all the mutations that their mother cells had. The mutations that you can detect, the somatic mutations you detect when you sequence a tumor, are ones that have been deposited in the ancestral lineage of that cell all the way back to the embryo, right? And so if you think about this, the single cell, kind of you think about the coalescence, you think about the cell going backwards in time, accumulating all these mutations, and we distinguish between two types of mutations, these so-called driver mutations. These are the ones that provide some sort of selection advantage, provide these hallmarks of cancer. They either lead to proliferation or lead to immune evasion, some other capacity or phenotype that the cells require. Because they provide selection advantage, they define lineages or epochs. They increase the frequency of cells that have those mutations. And by doing so, they also increase the frequency of mutations that were already present in the genome when the driver mutation was acquired. Okay? And so these other mutations are so-called passenger mutations. Great. Okay. So now the thing about these driver mutations, and this was kind of an issue that was discussed in the other talk, is we know what some of them are, but we don't know the functional significance of every single one of the mutations that can happen to a genome, right? And so the way in which driver mutations are detected is they seem to be overrepresented among tumor samples. This makes them rare and hard to find, and as I think should be clear by now, it's kind of a many-to-one problem in the sense that if you're going to break a gene or you're going to give a gene a capacity that it shouldn't have, there's many ways in which that can be done. There's many mutations that can lead to the same outcome, which makes it even more difficult to define these driver mutations. So people have been focusing on these driver mutations for decades. But now, with whole genome sequencing, we have the capacity to look at thousands of mutations, of which only a very small proportion are drivers, maybe 0.1% of them. And because they're so common, we can analyze these patterns of mutations as a way of trying to define cell state. And why is this important? This is important because we can analyze not only the mutations in the cancer, but if we can define them, we can analyze mutations that occurred during the progression from normal cells to cancer, to ask questions about potentially cell states that we can no longer observe. Okay? Why? And this makes sense from the point of view of someone who really wants to develop methods, because it's not entirely clear how to analyze these things, and there's a lot of them, right? So it's a perfect combination of things for people in methods development who don't want to spend a lot of time reading literature. Okay, all right, and just to kind of make this point a little bit more strongly, this is something called a Vogelgram, which you see. And it's one of the most well worked out genetic models of progression from like a normal colonic stem cell to like a cancer, a carcinoma. And so it has multiple stages, and each one of these stages is marked with pathways which indicate driver mutations. Now this sort of so-called Vogelgram has evolved over the years, and then initially it used to be like individual mutations, and now those mutations are grouped by pathways. And the reason it's been evolved in this way is because of this many-to-one problem, right? So we're talking about now a pathway, or maybe we're talking about a set of driver mutations that might have the same outcome. It's quite difficult to define those things. But if you see what's being shown here is we're seeing changes in cell state. right? And so if the passenger mutations that we're seeing in the cancer genome are accumulating in cells in different states, and those mutations can tell us something about, can give us some readout of those states, we might have a stronger way of defining these genetic changes in terms of phenotypic changes, because we're getting a direct phenotypic readout. Does this make sense so far? Great. Okay. So what do we have to do? Well, we have to identify these kind of different epochs and group mutations into them. Sometimes there's mutations that are going to be shared by all the cells, and those are mutations that happened before what's called the most recent common ancestor. That's the ancestor of all the cancer cells in the tumor. And there's going to be some mutations that are going to be only in a subset of the cancer cells. We're going to call those subclonal mutations. The ones that are shared by all the cells, we're going to call them clonal mutations. And so within this context, we have to do two things. First of all, we have to be able to group mutations into these epochs. And I'll talk a little bit about subclonal reconstruction, but I'm spending most of my time on the intro, so I'll sort of skirt through the methods, except to say that you can pull out a lot of different things in your methods toolbox to analyze different parts or different subproblems within this problem. So we've used Bayesian inference and Markov chain in Monte Carlo. We started exploring more discrete optimization strategies for work that I'm not going to talk about, but be happy to talk about in person. We're looking at matrix factorization, dynamic programming, and a cool new method called, has anyone heard of pruned exact linear time PELT? Has anyone heard of PELT? No one's heard of PELT? Okay, all right. I'm not going to tell you what it is, but you can look it up or ask me about it afterwards. But it's a great way to do segmentation. And so we're going to use matrix factorization and dynamic programming to segment things, and then we're going to use deep learning in a kind of a supervised setting. And each of these methods contribute to our ability to either group methods, mutations, and epochs, or infer something about historical cellular states from these somatic mutations that we're observing. Yeah?
So why is there information about the cell state that's encoded in somatic mutation?
Why?
Yeah,
Oh.
It's not expression. Just
Yeah. Great question. Why don't you ask that question at the end? Okay. If it's not been answered in the next 3 or 4 slides. Yeah. Okay. So, first of all, let's just talk about how this characterizing of the cancer populations is going to work. We take a heterogeneous sample of the tumor, so there's genetic changes among the cancer cells in the tumors, also normal cells. We're going to subject that sample to whole genome sequencing. The most common and widely used technique now is short read sequencing. And that's important because even though there's a lot of mutations in the genome, 5,000, 10,000, that's a big number, the genome's quite big. So because it's short read, most of the reads that we get are only going to contain a single mutation, right? And so we're going to try to define these things, both reconstruct the evolutionary history, which is represented by these epochs and their relationships to one another, and we're going to try to say something about the mutations just simply based on 2 types of information: what mutation occurred and its allelic frequency within the population. Okay, those are the things that are easy for us to measure. Now, I'm going to focus primarily on those types of the point mutations, and what we can get from point mutations when we map multiple reads to the same locus is we get a count of the proportion of the reads that contain this. This gives us an estimate of the allele frequency. When you do whole genome sequencing, you can also reconstruct copy number variants. An important copy number variant for us is a whole genome duplication. And you can also reconstruct structural variants, but they're much more difficult to reconstruct. Right, because the structural variants require you to target a specific breakpoint, largely in order to understand where the variant occurred. Okay, so how do we group mutations into historical epochs? I'm sorry, I told you I'd answer your question in 4 or 5 slides. Let's say 7 to 10. Okay, all right. So how does this work? So we have 2 cells. This is our population sample. And these mutations here indicate, color mutations indicate different colors, different distinct somatic mutations. So you can see here both these cells have 2 copies of the green mutation. They both have 1 copy of the yellow mutation. The purple cell is a purple mutation, and this has those things. So some of these mutations you could call clonal because they're present in each one of the cells, and some of the mutations are subclonal because there's only 1 copy in each one of the cells. Okay. Now the problem, of course, is is before sequencing, you lyse the cells. We're not doing single cell sequencing. That's not quite a clinical standard yet. And now all we have is we have alleles. And so there's two big problems here. We don't know how many cells necessarily went into the amount of DNA that we have here, which makes it difficult for us to compute cellular frequency, but we can certainly compute allelic frequency in the population. Okay, so 4 alleles of green, 2 alleles of yellow, blah, 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 blah. Okay. Now, I posit, based on this information alone, we can already distinguish 3 or 4 different epochs, right? And so, presumably, if we're able to detect the fact that there's been a genome doubling event, well, we see that there's some alleles that have allele frequency 4, and there's some alleles that have allele frequency 2. You know, one explanation for that is that this green mutation happened before the genome doubling event. Now it's present in 2 copies per cell. The yellow mutation happened after the genome doubling event, but before the most recent common ancestor, and so it's present in 1 copy for each of the cells. And then these mutations that are less frequent are subclonal. Okay. So now with this information, we have now defined 3 or 4 different epochs, depending upon how you want to define the subclonal things. And you know, it turns out what I'm showing here is actually real data. So if you take like in this case a sample of pancreatic cancer, sequence it to an appropriate depth, then you just plot the proportion of mutations at an estimated number of mutant alleles per cell. You can see 2 or more peaks. One peak at 1 estimated average number of mutant alleles per cell corresponding to clonal mutations. 1 at 2 per cell corresponding to the clonal mutations that happened before the genome doubling event. And the subclonal peaks are at less than 1 mutation per cell. Okay. Great. Huh. All right. And we can further analyze these subclonal peaks. Now we've just zoomed in on that further peak, and we have 4 different peaks here. Each of these 4 different peaks corresponds to a different lineage, and they correspond to a different lineage because, man, I don't know what's going on with me today. I'm just not feeling it. Okay. Hi, everyone. So, do you accept the fact that I can define subclonal lineages by just looking at allele frequencies? Yeah? Okay. All right. All right. So let's do that then. Okay. So maybe very quickly, I'm going to say why this works. And the reason this works is if you were able to measure the allelic frequency of mutations perfectly, you would expect to see 4 different peaks here, 4 different delta functions, basically, where all the mutations, the one driver, presumably that define the clone lineage, and all the passenger mutations, these are hitchhiker mutations that were already present in the cell that acquired the driver, would be driven to exactly the same frequencies. Okay, that's because the driver gives some sort of selection advantage. It grows out. A subsequent population that contains the initial driver gets a new driver. It increases in frequency. It has descendants that gain additional advantages. They increase in frequency. And so at the end of the day, you have basically 4 distinct populations that were present at some point in the tumor's history. And you can define them by what proportion of the cells contain those populations. Right. Now, if we can measure allele frequency perfectly, we would see these spikes. We can't measure it perfectly because we're doing finite sampling, so we see some sort of spread. And if we just do clustering, we can identify where those spikes might have been. Okay? And so by doing clustering here, we say there's 4 subpopulations. There's not. There's 3 subpopulations in the cell, but we still detect the B lineage because even though there's no cells that contain only A and B and not C and D anymore in the sample, the B lineage is still represented because there's 2 distinct children that had it. Got it? Okay. And then we can take that information and build out some sort of clone tree just by using a no homoplasy constraint, which I'm not going to explain. But what I will point out is that uncertainty in clustering, so if we get the clustering wrong, then we'll get the tree wrong. because we'll estimate the estimated number of mutant allele frequencies incorrectly. Okay. There's value in doing this just by itself because increased subclonality, having more subclones, is associated generally with poor prognosis in cancer. So in this specific example, and this is work done in collaboration with people who are now at UCLA, Paul Boutros, they were able to identify using our tools that subclonality in a small prostate biopsy is associated with twofold increased risk of relapse. Okay, so back to your question, how does mutations tell us anything about cell state? Well, I think the first thing we have to define is, well, what is the relationship between mutations or patterns of mutations and cell state? Okay, so the first way that relationship works is through the analysis of mutation spectra. Who's heard about mutation signatures here? Okay, good. All right. I love this stuff because this is new and cool. Okay, so this is what's called a mutational spectra of a breast tumor. This is a mutational spectra of a lung tumor. Okay, and so what is this mutational spectra? So there's 96 bars here. This is a histogram, and it's a count of 96 different types of mutations. So what's a mutation type? Well, a mutation could be like an A to a C or like a C to a T. So there's 6 different types of mutations that you can distinguish because you don't know what strand a mutation occurred on. And then whether or not a specific base is going to be mutated is also influenced by the 5' base or in the 3' base, right? And so 6 times 16 is 96, and those are the 96 different mutation types. Now, if you just count the number of mutations of each type in a tumor, and this is in this, I think, is from whole genome sequencing. So this is percentage mutations of the type. You can see there are distinct differences between mutational spectra. And this is a little bit like palmistry. So I can look at this picture right here, and I can tell you that this mutational spectra, this tumor sample, is probably taken from an older woman. I can tell you this tumor sample was taken from a smoker, someone who smoked or at least early on. And then there's been some intracellular process which has either been associated with inflammation or has led to an increase of mutation rate in that cell simply because that process is acting inappropriately. And I can do that because specific peaks in these mutational spectra are associated with different mutational processes. So in this case, these 4 peaks right here, 1, 2, 3, 4, are like the lifeline. And these mutations are almost like clock mutations, and they accumulate at a consistent rate with age, with wall clock time, if you're a computer scientist. Okay, and these mutations, the C to A mutations, they are associated with tobacco mutagens, right? These mutations here, these 2 peaks, are associated with a process called with the apochs enzymes. Okay, and the reason we know this is that about 6 years ago, someone in Peter Campbell and Mike Stratton's lab, Ludmil Alexandrov, just did non-negative matrix factorization on tens of thousands of mutational spectra looking for patterns of co-occurring mutations. And these co-occurring patterns of mutations, which are indicated here and called mutational spectra, and there's not only the patterns of co-occurrence, but their frequency of co-occurrence. Many of these mutational spectra, and they initially defined about 30 of them, could be immediately associated with either various different types of carcinogens like smoking or UV light, or could be associated with certain types of DNA damage or inefficient repair of DNA damage, so like MMR. This is sort of like a DNA damage repair type thing, apochs, and some of these patterns are associated with mutations caused by specific treatment. Yeah.
So these histograms from previous slides, they characterize a person at some stage in life?
They're taken from the tumor. So they're patterns of somatic mutations that are present in the cells, and presumably they describe the mutations that accumulated either during transformation from the normal cell into the most recent common ancestor of the cells, or mutations that accumulated after that transformation.
So you would have, for every tumor, you would have different histograms?
Yes, every tumor has different histograms, but there are common patterns of co-occurrence of mutations. And so that's what these signatures represent. as they represent these patterns of co-occurrence of mutations. Thank you for clarifying that. Yeah, and you can detect them using a technique called non-negative matrix factorization.
Can you cluster these histograms?
So we don't. I'm trying to think if people have. They might have. I mean, this, non-negative matrix factorization is kind of like clustering, right? And so it's one way of thinking about it is if you have a profile and you want that profile to belong to multiple clusters, you could use something like non-negative matrix factorization in order to detect those kind of joint cluster memberships. There's another dimensionality reduction technique called independent components analysis. You can kind of think of it as allowing occasional multiple clustering.
Please continue and we will discuss that later.
Okay, so this is one way in which cell state can reflect what the type of mutations that you see. Another way cell state can reflect the type of mutations that you see is that, at least for some types of mutations, they accumulate in closed chromatin. And so here, this is, has anyone seen this picture before? No? Two people? Come on, there's more of you. Okay, so does everyone know what chromatin is? Chromatin. Chromatin's good, and somatic mutation's good. Okay. I bet you didn't know that they were associated with one another. So this black line here, this is the density of mutations, and this chromosome, which is chromosome 2, has actually been chunked up into 1 megabase regions. And this is just the density of C to T mutations that are accumulating or that were detected in a few dozen melanomas. Okay, melanomas have a lot of mutations, and so we get pretty good statistics on the mutation density within specific regions of chromosome 2. Okay, the blue line is a measure of chromatin closedness in melanocyte. Melanocytes are the presumed cell type of origin for melanomas, right? And so, as I hope you can see, there's higher mutational density wherever there's more closed chromatin in melanocytes, right? And this is a pretty powerful observation, right? And so this is not true of all mutation types, and the relationship between chromatin state and the source of mutations does vary among mutation types, but at least in this specific case, these mutations, which are caused by UV light, seem to accumulate in closed chromatin. It's not entirely clear what the reason for this is yet, but the most commonly reported reason that I've heard is that there's inefficient repair of UV-caused mutations in closed chromatin. There's more efficient repair in open chromatin, right? But this is a very powerful observation because what that says is, because chromatin state varies between cells, is that you can construct something about cell state by looking at mutation density during different historical periods. Okay. So we know that cell state affects where somatic mutation is going to accumulate, and specifically both the mutation source and the chromatin state affect mutation type and genomic location. Okay. So here's the question: can we go backwards? And that's where I think the key interesting methodological development is already happening and can happen in the next 3 to 5 years. Okay. There's no way for me to know about time, but I felt like I took a really long time with my intro. So where am I?
9 minutes.
9 minutes? Okay. I got three things I can talk about. All right. I can talk about how to define these epochs. That's number one. We're going to have a vote. I can talk about how to go from genomic location to tumor type. I think that's the one I'm most excited about talking about, but I'm also excited about the other things. And number three, I'm going to talk about how to reconstruct a historical record of changes in mutation source by looking at a single sample of a tumor. So we're going to have a vote. Vote for number one. One. Okay, vote for number two. Vote for number three. I'm so glad we're excited about the same things. Okay. Here we go. Okay, so we spent a lot of time thinking about how to reverse this problem, right? So go from mutation density at historical periods and reconstruct things about cell state. Now, the thing to keep in mind is the pictures that we saw, they were generated using millions of mutations, right? So to get density estimates that good on a tumor-specific way, you're just not going to get that because you have like 5,000 mutations. And so before we even started, the biggest question is, well, how much information is there even in the mutation density? Okay, so if I take the mutations from a single tumor, like 5,000 on average, and I group the chromatin, chunk up the chromatin in 1 megabase regions, I have 3,000 bins. And so on average, I'm going to get less than 2 mutations per bin. How much information does that tell me about the cell, even the cell type of origin of the tumor type? So to answer these questions, as part of our participation in a consortia called the Whole Pan-Cancer Analysis of Whole Genomes Project, or PCAWG, which was part of both the ICGC and the TCGA, we joined a group of 800 researchers who were divided into about 16 different working groups to jointly analyze whole genome sequencing data to about 35x on average for about 3,000 tumor normal pairs. And we wanted to ask the question, if we just look at these patterns of somatic mutations, could we recover what the tumor type is? I think it's a very weak type of information, but it's, even before we start, that's the first question to ask. And so here I'm showing here is just the relative distribution of donors of different samples. So we have a lot of pancreas tumors, samples of pancreas cancer, liver cancer, various types of brain cancer. Okay, and so we looked at 2 types of mutation patterns, and when I say we, I mean Wei Zhao, who is a student, who's a data scientist and Lincoln Stein student and lab at the OICR, and he used to actually be a student in my lab, and Gurneet Atwal, who's a current PhD student in my lab. And they define 2 types of features. One is just the counts of the number of mutations in 1 megabase regions as you go across the chromosomes. And the other one is these SMV types, so this mutational spectra. And the question is, how predictive of these various things we want to predict about the tumor? First of all, just even tumor type. And so Gurneet used deep learning to predict tumor type. When you have a lot of data and you don't really understand the relationship between the input and the output, deep learning is a fantastic thing to do. And so one thing that I'll say about this is if you do want to do deep learning, you have to optimize your hyperparameters. Does everyone know what a hyperparameter is? Yeah, okay. If you don't optimize your hyperparameters, so parameters are things that are the parameters of your model that affect how the input is mapped to the output. Hyperparameters are things that tell your method how to train the model or describe properties of the model. So in this case, our hyperparameters are the deep learning architecture. How many units per layer, how many layers to have, what the relationship is between the input to a unit and the output of the unit, and also how we train the model. And so in order to actually get the performance that we did, we actually had to optimize these parameters. And so skopt, if you're going to learn one thing, you should learn that collection of 5 letters, because that's going to make your deep learning work a lot better. Do you have a question, Ben?
No, I'm just learning.
Okay, all right. And so Gurneet did all this, and then he trained, it turned out, an ensemble of methods because it's actually an easier way to optimize your hyperparameters for reasons I'm happy to tell you outside of this talk. But so he trained his method, but before we did this, Wei was even asking a simpler question. And Wei's question was, let's say if we trained a method on the patterns of somatic mutations, which are primarily passenger mutations, or we took the driver mutations. So this is like all the history that has been defined in the relationship between specific somatic variants and tumor types, which would be the more predictive set of features. Okay, and so Wei, to do this, he trained a random forest, and here are the results. Each one of these points corresponds to a different tumor type. There's 24 different tumor types. The F1 score is a measure of the accuracy. You can think of it as a measure of the accuracy of a classifier. It's the harmonic mean. Oh, sorry, mutations, right? Harmonic mean between the precision and recall of the classifier. And he trained a random forest. And so this is how well you perform if you just look at the mutation spectra augmented with some dinucleotide features. This is how well you can do if you look at the distribution among these 3,000 different 1 megabase regions. And this is how you do if you take a vector which describes whether or not the tumor contains a set of about 500 different driver mutations that either came from the literature or defined by the driver group within PCAWG, which is, you know, some of the world experts on driver mutations. And as you can see, these passenger mutation features, they perform much better than driver mutations. And so, you know, it's very easy to see why this is the case. So for example, it's rare, there's incomplete knowledge about driver mutations. So in fact, a quarter of the tumors that we had didn't have any drivers defined for them at all. The other thing is that driver mutations, they're not always unique to different cancer types. A lot of cancers have P53 mutations. All right. There are some that are unique or pathognomonic, but they're relatively rare and they're not present in every tumor of that type. Okay. Now, if you combine the SNP type and distributions together, and then you train this deep neural network in the way that Gurneet did, using Bayesian optimization, we get much better performance, as you might expect. And then when we add drivers, it doesn't add to the performance. And in fact, it decreases the performance a little bit because we have, like, you know, more than 3,000 inputs, but we have less than 3,000 data examples. So we imagine there'd be like a tiny bit of overfitting, though obviously some of the hyperparameters that we're fitting have to do with regularization. And, you know, the classification errors that we're seeing, they reflect the tumor type. So this is what's called a confusion matrix. And so the rows here tell you what the correct answer is, and the columns tell you what the predicted answer is. And if you're on the diagonal, this is the proportion of samples where the predicted answer is the correct answer. And the off-diagonal elements tell us about frequent misclassifications. So we frequently misclassify stomach to esophageal cancer. In fact, this misclassification happens a lot clinically as well. There's a lot of cancers that are near the border between the stomach and the esophagus. So it's actually hard to make this classification even for clinicians. Other things are like we confuse different B-cell malignancies, which is not surprising. What is surprising that we can actually distinguish them to some degree. Now, the great thing about, people have done this with like RNA before, and they've tried to do it with like driver mutations. And, you know, the performance we get, while pretty good, is, you know, comparable with the best of these methods. But the thing that's nice about looking at somatic variants is they don't go away. So we took the same classifier, we took data from a completely different study where they sequenced 1,500 metastatic tumors. Now in this study, one thing that we learned about being members of the PCAWG is somatic variant calling is not perfect, and there's a lot of variability between different methods in the somatic variants that they call. So this metastatic data, which came from a variety of groups in the Netherlands, they used a different pipeline. So they had different somatic variant calls, and these sequence metastases rather than primaries. And despite those two things, we could actually get classification 82% accuracy, which was quite good. We still made the sort of mistakes that you would expect us to make. But this type of classification is a quite difficult problem for clinicians to solve. So when they're asked to solve a similar problem using histopathology, their accuracy in distinguishing between about 11 different types of cancer is about 50%. Okay. All right. So what does this mean? So what this means is that at least we can go from mutation type and genomic location to tumor type. Right? There is enough information there for us to kind of reverse this. And what I didn't show you is that you can divide mutations that you detect in a tumor sample into different historical eras just by looking at changes in the perceived mutation source. So not only can we say something about tumor type, we can also do this kind of historical reconstruction. What we don't have now is we don't have the ability to go from these things to some sort of measure of change in chromatin state. And that's what I think is the big, open, and interesting problem in this field. And certainly I think to solve it, oh, I didn't have this slide. One way to solve it is to use all the information that we have. So not only genomic location, but tumor type and allelic frequency. And people really haven't started to combine all those three different information modalities together. Also, there needs to be some sort of latent modeling of a dynamical system which describes the transformation from the normal cell, where we understand what the chromatin state is, to the tumor, where we can actually measure the chromatin state and try to fill the things in between based on the mutation densities. Okay, so I'd like to acknowledge the various funding bodies that funded our work and the people who did the work in my lab. I focused on work for people whose names are bolded. The work on the deep learning for tumor type that was done in collaboration with Lincoln Stein, the OICR, and Paz Polak, who's now on Mount Sinai, and the groups in the Netherlands that did the sequencing of the metastatic samples. The people here were members of the PCAWG group that I was a member of, and these are the people who provided funding for our work. And if I have any time left, I'm happy to take some questions.